Hello everyone, myself is Dr. Avinash Amangi. I am from Gurudev Academy, Dharwad. So we are going to discuss here the biology part of the particularly NEET 2025, right? As I am your biology mentor, so today we will discuss with what NEET question paper and that to what mainly biology part of it is, right? So we'll go ahead with a question here, right? So majorly the complex 2 of mitochondrial electron transport chain is also known as, right? So they are asking related with what complex 2, right? So majorly you options are given here are the cytochrome BC1, then succinate dehydrogenase, cytochrome C oxidase and NAD dehydrogenase. So with respect to the electron transport system, if I consider, right, so which will actually take place with respect to the inner mitochondrial membrane, right? So if I see the structure of what mitochondria, inside the what majorly like what this cristae, where inner membrane of what mitochondria will carry out what foldings. With respect to that only this electron transport system is going to take place. So here these electron transport systems are divided in different complexes: complex 1, complex 2 and even what the complex 3 and complex 4, right? So what we know is the first one will be the what complex 1, right, where whatever the NADH formed, right, NADH2 or whatever this NADH is formed, that is actually going to come and enter inside what the complex 1, right? So that will enter with respect to the what here complex 1 and donate something called as what electron, right, and gives back to us what something called as what 4H plus ions, right? So here mainly this NADH dehydrogenase will come, right? So which actually what reduces this component, right, and releases what electrons inside the particularly electron transport system, gives back as something called as what proton, and that is what with respect to the what complex 1. So in between of complex 2 we are going to get complex 1 and complex 2, we are going to get here what the complex 3, right? And with respect to the complex 3, I'm going to get here majorly the cytochrome BC1, right? So what we are going to get here the cytochrome BC1, right? Now here the complex 2 if you consider, right, complex 2, from the complex 1 whatever the electrons are that will move towards what complex 3, right, and from the complex 2 whatever the electrons that will move towards what complex 3 again, right? So there we'll find out what the presence of this particularly cytochrome BC1, right? This is cytochrome BC1 with respect to that majorly like cytochrome C, right? Cytochrome C with respect to the what complex 3, cytochrome BC, cytochrome C to what C1 to what C2, it is going to what release the electrons, right? So like that of what a transportation we are going to see. So from the complex 4 it will enter inside what the complex, sorry, from the complex 3 it will enter into the what complex 4. So they asked us related with what complex 2. So there whatever this particularly what succinate which got converted into what the fumarate, right, in presence of an enzyme succinate dehydrogenase, in presence of what enzyme succinate dehydrogenase, right? Then with respect to the what the complex 4 if I consider complex 4, what I am going to get here, that is nothing but what the cytochrome C oxidase, right? With complex 4 I am going to get what cytochrome C oxidase. So according to the question if I see, what I am going to get the complex 2 of a mitochondrial electron transport chain is also known as what here the succinate dehydrogenase. What it can be known as succinate dehydrogenase, that is what here I have mentioned complex 2. Complex 2 consists of what here? The succinate dehydrogenase enzyme. Alright. So with respect to this question here, the option 2 is what correct. Right. Then moving on to the next question, that is polymerized chain reaction, that is what PCR, amplifies the DNA with the following equation. Right. Now what we know is, consider I have a less amount of what DNA, right? Then I'll go ahead with what? The amplification of this DNA, right? Amplification of DNA. And this amplification of the DNA can be carried by means of what? PCR method, right? That is what we call it as polymerized chain reaction, right? So which is actually to be carried out inside what? A machine called as what? Thermal cycler, right? So there majorly like what? First step with respect to the PCR, we are going to see what? Denaturation of the DNA, right? Then second one is known to be the what? Extension, right? uh, Sorry, not the extension, but here the second one, annealing procedure, right? Annealing to what? The particularly extension. So what I am getting here, the small segment of the DNA which is actually what? A denatured, right? Now that will be attached with what? A primer by means of what? Annealing, and then the overall extension is going to take place. So with one cycle I'm going to get what? The two copies of what? The DNA from 1 to what? 2. So obviously I'm going to use the formula here, what? 2 raised to n, right? The formula I'm going to use is what? 2 raised to n, right? And that will give me the million. After the completion of what? 30 cycles, I'm going to get million copies of the what? Same DNA, right? And that what actually the PCR stands for, and that we call it as amplification of this particularly DNA, or making or increasing the number of what? Copies of this particularly what? DNA, right? So answer here is what? That is 2 raised to n, right? Related with what? 92 question, right? Moving on to the next, what are the what are the potential drawback in adoption of the particularly what? IVF method, right? So as it is related with what? Potential drawback, what I am going to get it with respect to the IVF. IVF means it is known to be in vitro fertilization, right? So we are going to carry out the fertilization in the laboratory scale, and that itself what we call it as IVF, in vitro type of what? Fertilization. So they have given something called as what? Different options, majorly like what? They asked us potential drawbacks from this, right? So if you consider here, what they are saying, a highly fatality risk to the what? Mother, that is what? Option A. Then B says expensive instrument and reagents. Then C says what? Husband or wife necessary for what? Being a donors, right? Then D says less adoption of what? Orphan. And E says what? Not available in India, and possibility that the early embryo does not survive, right? So according to that, by seeing what? The choose the what? Correct answer over here, we have to find out the answer with respect to that, that is choose the correct answer from the what? Given option. So according to that, if I consider here, that is mainly with respect to the IVF, if you consider what is actually the potential drawback here, that is expensive instruments and reagents, right? So it requires what? Expensive instruments and even the reagents. Then you consider what? Less adoption of what? Orphans. Now what will happen if we go on carrying out with respect to the babies are produced with in vitro fertilization? Definitely there are what? Couples which will not go for what? The adoption of what? Orphans, right? So orphans will be what? Left there itself, right? Then you consider possibility that the early embryo does not survive, right? So early embryo, after the what? Fertilization you consider in vitroly, so early embryonic what? Death can takes place. So that I am going to see. So according to these options, the potential drawbacks are what? These three of them. So that is option 1 is correct, that is B, D and F only, right? Then moving on to the next question, that is what is the name of the blood vessel that carries deoxygenated blood from the what body to the heart in the particularly what frog, and that is nothing but what the vena cava, right? What we are going to get over here is the vena cava, right? So the blood vessel that carries deoxygenated blood from the body to the what heart, body to the what heart in particularly what frog, that is nothing but what here the vena cava, right? Vena cava itself is going to carry out, right? Vena cava itself is going to carry out what? Deoxygenated blood, deoxygenated blood majorly from the body, majorly from the body to the what heart, right? And that is with respect to the frog, right? And answer is here the vena cava, right? Answer is here what? The vena cava, right? Moving on to the next. Which one of the following statements refers to the reductionist biology, right? So reductionist biologist and the statement related with him, right? So physicochemical approach to the what study and understanding the living organism, physiological approach to the study and understand living organisms, chemical approach to the what study and understanding living organisms, behavioral approach to the study and the understanding what living organisms. So these are the what the four options what they have given. So based on that, reductionist biologist, if you consider, they are going to understand majorly the physico chemical approach, right? Or physical or chemical type of what reaction which can take place with respect to the what living organisms, which are majorly going to take place with whom? The living organisms. So those concepts are to be understood by means of whom? This particularly what reductionist biologist, right? So here the option is what? One, right? As simple as that, right? Then moving on to the what next question, right? That is they have given one kind of what statement here. That is statement 1 says in the RNA world, RNA is considered the first genetic material evolved to carry out essential life processes, right? Then they are saying RNA act as a genetic material and also as a catalyst for what? Some important biochemical reactions in living system. Being reactive, RNA is unstable, right? Then they are saying what? DNA evolved from RNA and is more stable genetic material. Its double helical strand being complementary and resists the change by evolving repairing mechanism, right? So according to this particularly, we have to find out what which statement is correct or which statement is incorrect. So these both the statement if I consider, we have already discussed with respect to the what molecular basis of inheritance related with what RNA. The first genetic material to be considered was what? The RNA, and from the RNA only who got evolved? The particularly DNA. Understanding? So and that itself known to be the what? The particularly RNA act as a what? Genetic material. In most of the viruses you consider, it can act as a genetic material, and also it catalyzes some of the important biochemical reaction. And as RNA carries the catalysis of what biochemical reaction, we majorly call it as a ribozyme. We majorly call it as what? Ribozyme here, right? So which initiates something called as what? The biochemical reaction, majorly which will be with respect to the what? rRNA, which will act as what? Ribosome with respect to the ribozyme with respect to the what? During the protein synthesis, right? In protein synthesis, it is going to form a peptide bond between the two amino acids, right? So there, so according to this, stability wise if you consider, RNA is unstable molecule. As we know, the molecular formula of RNA is what? The mainly pentose sugar C5H10O5, right? And for the DNA it is C5H10O4, right? So one oxygen is less inside the DNA. So at the what? Second position of what? This particularly pentose sugar, second carbon position of pentose sugar, there will be one oxygen more and which will form what? This hydroxyl group inside an RNA. So only what it became the unstable. So it can react with respect to the what any of the component, right? So like that of what the component what you can consider. So here the statement 1 is correct. Statement 2 if you consider, it is what the DNA, if you consider DNA evolved from where RNA only, and is more stable genetic material, because what we know, one less oxygen what we are going to find out again, where with respect to the pentose sugar at the what second position of what this carbon, there will be what only hydrogen group, right? So there will be an absence of what this particularly oxygen, right? And that makes the DNA as a what a stable material, stable genetic material, and it has what resist and change by what evolving and repairing. So here the particularly statement 2 is even correct. So according to that, if I consider here, both the statement 1 and statement 2 are correct, right? That is what majorly option 1 is correct. Moving on to the next, that is what epiphytes that are growing on a mango branch is an example of which of the following, right? So you have already studied related with what organism and population. Inside that we have came across the population interaction. So with respect to the population interaction, if I consider here the orchid majorly, which is known as what epiphyte, orchid which is known as what the particularly epiphyte, the organisms or the plants which are grow onto the what other plant, and this epiphyte grows onto the what actually the mango, right? Mango plant, right? Now it is going to take all the benefits, right? Who is going to take all the benefits? Orchid is going to take all the benefits from the what mango, right? But it is not going to harm this particularly mango, right? But mango is not benefited by means of what orchid. So only what we call it as, it is this kind of interaction is actually known as commensalism. This type of interaction is known as what? The commensalism, where it is an interaction between what two different species. It is an interaction between what two different species. One is benefited, another one is neither harmed or nor benefited, right? Neither harmed or nor benefited. That what we call it as commensalism. So here the answer is what actually commensalism. In mutualism, what we are going to see? We are going to see both of the organisms are benefited from each other, right? So example you consider lichen. Now predation what you are going to find out? With respect to the predation, one of the strongest animal is going to feed onto the what weaker organism, right? So predator will feed onto the what prey, right? So that is what you consider lion and deer or tiger and deer, right? So that is what the predation. Amensalism, where one of the organism like kills the other organism, but here it is not benefited or nor what it is harmed, but it is going to kill the other organism. That is what we will find out with respect to the amensalism, right? So in amensalism example you consider Penicillium notatum growing inside a bacterial culture. So Penicillium notatum will kill what the bacteria, but after killing it is not what benefited. Who is not benefited? Penicillium notatum is not benefited. So according to this particularly question, if I consider, answer is what? The commensalism. Answer is what here? The commensalism, right? So example based what we have to remember here, right? From the statement given below, choose the what correct option, right? So they have given some statements, that is eukaryotic ribosomes are 80s and prokaryotic ribosomes are 70s, right? Then each ribosome has two subunits. Then what they are saying? The two subunits of 80s ribosomes are 60s and 40s, while that of what 70s are 50s and 30s. The two subunits of what 80s ribosomes and 60 are what 60s and 20s. 70s has what 50s and 20s. The two subunits of what 80s and 60s, that is what 60s and 30s, and that of what 70s is 50s and 30s, right? So if you consider with respect to the what structure of what ribosome, right? Ribosome structure, if you see, it is actually known to be the suborganal, right, which has one unit known as what, larger subunit, right, which is larger with respect to the what, density even, right, and one more is what actually known to be the smaller subunit. It is known to be the smaller subunit here. Understanding? So now when I compare with respect to the what, eukaryotic organism, right, inside the eukaryotic I'll get what, 80s type of a ribosome. But when I see with respect to the prokaryotic organism, I'll see 70s type of a ribosome here, right, as combining both the subunits, as combining what, both the subunits structurally, like how it is the larger and the particularly smaller, right. But if you consider, when I say 80s type of a ribosome, it has larger unit with what, the particularly 60s, and smaller subunit is with what, 40s. When I calculate both of them, 60 plus 40 is actually 100, right. When these particularly are larger with the subunits, or what, they are separated in form with respect to the what, cytoplasm, during that period their density is more, right, and a smaller subunit even the density is more. But when it get joined together, right, that time it makes what, actually the 80s, not what, 100s, right. But even with respect to the what, prokaryotic, if I consider, it is of what, the 50s and 30s, right. So 50s and 30s makes 80s, but here it is what, having a 70s type of what, the ribosome. The reason for, when both of them are going to get combined, during that period the density will come down with what, different with respect to the what, in calculation too, right. So S stands for here what, actually the Sedberg's unit, right, and that is based on the density of this particularly what, ribosome. So here if I consider, the statement A is correct, right, then the statement B is correct, then the statement C is correct, but statement D is incorrect, because what they told is 80s is of type of what, 60s and 20s they are saying, but it's not 60s and 20s, it is 60s and 40s, right. 70s they have mentioned what, 50s and 20s, right. So it is not what, 50s and 20s, it is 50s and 30s type with respect to the word prokaryotic. Again here also they have made a mistake. 70s they have mentioned what, actually the correct, that is 50s and 30s, but 80s they have mentioned what, 60s and 30s, but it is instead 60s and 40s. So here this statement is even incorrect. So according to that, A, B and C are what, true. A, B and C are true. Here the option 1 is what, correct. Here the option 1 is correct, right. Then moving on to the next question. Which one of the following is an example of what, ex situ conservation? Which of the is a following example of what here, ex situ conservation? So you should know like what, ex situ conservation means, and ex situ type of what, conservation here, right, and one more is in situ type of what, conservation, right. These conservations are actually made for the what, maintenance of what, different species here. So here we made what, the maintenance of what, different species into particular geographical area. Consider I am maintaining the different types of species inside their own habitat, right, inside their what, own habitat. That's what I have called it as in, right, inside their own habitat. If I am carrying out, it means what, I am preserving them into the what, into their specific habitat where they have born, right. Majorly you consider like jungles, there we are going to preserve what, the different species. Such kind we call it as what, in situ conservation. And one more type is what, ex situ conservation here, right. In ex situ conservation, what we are going to do, man-made geographical area, there we are actually going to preserve an organism, right. That is what we call it as ex situ type of what, conservation. So according to that, option wise if I consider, national park and wildlife sanctuaries, these both of them are in situ conservation. protected areas means what, into their own areas only we are going to carry out protection. So definitely it is also what, in situ conservation. What left is zoos and botanical gardens. What left is what here, zoos and botanical garden, where we are majorly going to preserve the organism by creating something called as what, specialized settings, right? And those specialized settings are known to be the what, zoos and botanical gardens, right? Those specialized settings are known to be what, the zoos as well as what, botanical gardens, right? So here according to that, if I consider option 3 is what, correct, right? Option 3 is correct. Then moving on to the what, next concept, that is what, given below are two statements. That is, statement 1 says the primary source of energy in an ecosystem is a solar energy, right? So primary source of energy in an ecosystem is a solar energy. Then statement 2 says the rate of production of organic matter during photosynthesis in an ecosystem is called as net primary productivity, right? So as we know, sun itself is being what, the primary source of what, energy. The sun is trapped by means of what, plants, by means of what, the process known to be what, photosynthesis, right? And it produces, with respect to the photosynthesis, what it can produce, it produces an organic matter, right? It produces here organic matter. Now when I see this particularly organic matter, that organic, primarily whatever the organic matter formed is, that what I am going to call it as, I am going to call it as gross primary productivity, right? I am going to call it as what, gross primary productivity, right? Now this gross primary productivity with respect to the what, plant only it is produced, and plant is going to utilize some of the energy component from there, right? And during that period what will happen, this gross primary productivity is utilized where, with respect to the what, respiration, right? Means it is going to utilize that particularly energy. So here GPP minus R will give me something called as what, NPP, that is what, net primary productivity, right? So according to these particularly statements, if I consider, the first statement is what, true, that is what, the primary source of energy in an ecosystem is solar energy. What it is? The solar energy. Statement 2 says what, the rate of production of organic matter during photosynthesis in an ecosystem is called as, actually is called as gross primary productivity. It is called as gross primary productivity. Whatever the productivity left out, that I will call it as net primary productivity with respect to the whom, plant. Understanding? Left out form of what, organic matter, and formed by means of photosynthesis, the primary one, that I will call it as gross primary productivity. So according to that, if I consider here, the statement 1 is incorrect and statement 1 is correct and statement 2 is what, incorrect. Here according to this, if I consider here, statement 1 is correct but the statement 2 is incorrect, right? Not this option, right? Option here is what, 3, that is statement 1 is correct but the statement 2 is incorrect, right? Then moving on to the next question, right? So here we have to like what, match the following, some of the what diseases they have mentioned and which are related to what, right? So that what we have to find out. So according to that, if I consider here majorly emphysema is related with what, actually the respiratory type of diseases. Majorly emphysema even can be caused by means of what, smoking, right? So if I consider emphysema, it actually damaged alveolar walls and decreased what, respiratory surface, decreased what, respiratory surface, that is what emphysema, right? Then you consider angina pectoris, right? So angina pectoris is related with what, the acute chest pain when not enough oxygen is reaching to the what, heart muscle, right? Acute chest pain we are going to get, right, when oxygen is not going to reach our heart muscle, right? So that is what angina pectoris. Then glomerular nephritis, right? So glomerular nephritis is related with whom? The inflammation of what? Glomerular inflammation of glomeruli of what? Particularly kidney, right? As we know, kidney's basic unit is a nephron, and nephron is consist of what? Glomerulus filter, right? So there it is going to carry out and the particularly inflammation. Now when there is an inflammation, definitely it causes what? The particularly glomerular nephritis, right? Then tetany is related with what? Rapid spasms in the particularly muscles due to the what? Low calcium level in the body fluid. If a body fluid will have a what? Less calcium level, definitely we are going to get what? Spasms, right, with respect to the what? Our muscles, and that what we call it as tetany, right? So here according to this, if I consider, option 4 is what? Correct, right? Option 4 is correct. Then moving on to the next question. So given below are what? Two statements here, right? One like what? Assertion they have mentioned, and one is what? Reason here, right? Here what assertion says? Both wind and water pollinated flowers are not very colorful and do not produce what? Nectar, and they do not produce what? Nectar. Reason says the flowers produces what? Enormous amount of what? Pollen grains in wind and water pollinated flower, wind and water pollinated flower, right? So with respect to that, if I consider statement wise, if I go with respect to the what? Wind and water pollination, right? Now flower is not going to attract something called as what? Wind and water by having what? Colorful or even colorful or fragrance, or even what? By producing a nectar. So those colorful and being what? Nectar formation can be carried for what? The particularly insect pollination, right? For what? The particularly insect pollination. But with respect to the wind and water, it's not necessary actually. But for the wind and water, what is actually necessary here? That should be what? The amount of what? Pollen grain should be what? Formed will be what? More in number, right? Because due to the what? Flow of wind and water, some of like majority of the pollen grain will be what? Lost, right? So because of that only it produces what? Large amount of what? The pollen grains, right? So here if I consider, both the assertion and reasons are true, but reason is not actually the correct explanation of what? Assertion. Now flower and with respect to the wind, if I consider, they are saying what? Pollination related here, but here they are saying it is related with respect to the pollen grains, right? So which are what? Actually not related. So because of that only both assertion and reasons are true, but reason is not what? Actually the correct explanation of what? Assertion here, right? Then moving on to the next, which of the following is an example of what? Non-distilled alcoholic beverage produced by yeast, right? Which of the following is an example of non-distilled alcoholic beverage produced by yeast, right? So they are saying here non-distilled, right? Non-distilled type. And non-distilled type of what? Alcoholic beverages which is actually produced by yeast is beer and wine, is what? The beer and wine, right? But the distilled form of what? The particularly beverages, right? Distilled form of what? The beverages, they are nothing but what? Whiskey, right? Then you consider brandy, right? And even the rum. So only why distillation is required? Percentage wise if I found out, whiskey, brandy and rum will have a more amount of what? Alcohol percentage, right? So approximately you consider what? 45% of what? Alcohol what we are going to get, right? But with respect to the beer and wine, there the particularly we are going to get what? Less amount of what? Mainly like alcohol, that is what? 8 to 12%. 8% I can find out with respect to the beer, and 12% I can find out with respect to the wine here, right? So that is what. So here with respect to the what? Non-distilled alcoholic beverages produced by yeast are, so the whiskey is distilled one, brandy is distilled one, rum is even distilled one, but the beer is non-distilled. So option 3 is what? Correct here, right? Then moving on to the next. Then given below are what? Two statements, that is what? In floral formula that is what the plus and the circle symbol stands for zygomorphic nature of the flower, right? And G below dash, that is what gynoecium below dash stands for what inferior ovary they are saying, right? Then statement 2 says in floral formula H plus, sorry, this plus and circle stands for actinomorphic nature of what flower, and G dash stands for what superior ovary, right? So with respect to the what light of what above statements, what we have to choose both the statement and means what we have to see which is the correct and which one is the wrong, right? So as there are two types of flower, we consider zygomorphic and actinomorphic, right? So for zygomorphic I'll use actually the percentage symbol, right? For the zygomorphic I'll use what the percentage symbol, and the arrangement of ovary onto the thalamus, based on that only I am going to call it as superior or inferior type of what ovary, right? So if I consider inferior type of ovary, I'll give dash which is above the G, right? That I'll call it as what inferior type of the ovary, inferior ovary. But these both of them, if I consider, it is related with actinomorphic, right? And this below dash is related with the majorly superior ovary, right? Superior form of what ovary, understanding? So that is what with respect to the statement. If I can compare, this statement 1 is what incorrect, right? And the statement 2 is correct here. So according to the options, if I consider here, the statement 1 is incorrect and but the statement 2 is correct. That is what option 4 here, right? According to this, option 4 is what correct. Then moving on to the next, that is here the majorly like streptokinase produced by a bacteria, streptococcus, right? Streptokinase is a particularly enzyme produced by a bacteria known to be the streptococcus, is used for, right, is majorly used for here removing the something called as what clots from the particularly blood vessels, right? So streptokinase is an enzyme which is actually used for removing something called as what the clots from the what blood vessel, right? So there it is produced from the whom, the streptococcus bacteria. So here option 4 is correct. Moving on to the next, then which chromosome in human genome has the highest number of genome, right? That is what we are going to find out with respect to the chromosome 1. Least number of what the genes we are going to get it with respect to the what chromosome Y, that is what 231 genes I am going to get, and here I will get it like what thousands of genes you consider approximately, like chromosome 1 is what actually correct, where human genome has the highest number of what the human genes, that is what chromosome 1, right? More than that only what I am going to get, right? Then which of the following statement is correct about what location of what male frog copulatory pad, right? So male frog copulatory pad I am going to get it with respect to the what first digit of the what forelimbs, right? The first digit of the forelimbs, there itself I am going to form a particularly find the copulatory pad. So here itself the first digit of what forelimb I am going to get the copulatory pads here, right? Then moving on to the next, that is what which of the following phytohormone promotes the nutrient mobilization, right, which actually stores back the what nutrition, which help in delaying the what leaf senescence in plant, and that leaf senescence in plant I am going to find out with respect to the what cytokinin, right? Cytokinin which actually avoids the particularly leaf senescence or carries what a delay of what a leaf senescence, because it produces something called as what whatever the food is stored, right, or whatever the food is produced, it is going to what mobilize that particularly what the food material, who the particularly what cytokinin. So here option 4 is what correct. Then moving on to the next, that is while trying to find out the characteristics of a newly found animal, they have found one new animal and a research did the histology of the what adult animal. He carried out what the histology of this particularly. Histology means what? The particularly study of the tissues, right, related how actually the cells are arranged with respect to this. Observed a cavity with the presence of what mesodermal tissue towards the what body wall. What he observed? Mesodermal tissue towards the body wall, but no mesodermal tissue was observed towards the alimentary canal. He have not found a mesodermal tissues related with what? Towards what? Alimentary canal. What could be the possible coelom of that particularly what? The animals. So if I consider according to that, what I am finding out, it known to be the here pseudocloemate, right. So according to the options, if I consider, that is related with what? The pseudocloemate, right. Then moving on to the next, that is what? Match the least one with what? Least two. Match the least one with what? Least two here. So here they have mentioned what? Head, middle piece, acrosome and tail, right, which is actually related with what? Sperm. So if I see the structure of what? Sperm, it has what? Head. On to the head I am going to find out the cap, that is what mainly we are going to call it as acrosome, right. Then it has something called as what? Head, right. Head 2 I am going to find out what? Something called as what? Tail, right. So the in between of this I am going to find out what? Middle piece, right. So middle piece of this one. Then I am going to find out what? The tail, right. So according to this, I have to find out what exactly it can do, right. If I consider head, head is for what purpose? Either enzymes, sperm mobility, energy and genetic material. With respect to the head only, I am going to find out the genetic material being present. So here the head is related with the genetic material. Middle piece is related with what? Middle piece has what? Highest number of what? Mitochondria. Obviously mitochondria is meant for what? Giving an energy, right. So obviously middle piece is related with what? Energy. Then acrosome, if I consider, it consists of an enzyme, right. So which will actually help up in the particularly what? Carry out a fertilization, right. So that the sperm can get enter inside the what? Ova, right. And the tail is meant for what here? The sperm motility, right. Tail is meant for here the particular sperm motility. According to that, if I consider overallly, here the option 1 is correct. Here what? The option 1 is correct, right. So this is what I am going to find out, right. Then moving on to the what? Next question, that is what? Given below are the stages in the particularly what? Life cycle of what? Pteridophyte, right. And arrange the following stages in the correct sequence, right. So these are the stages of what? The particularly the pteridophytes, and what we have to do is we have to arrange it according to the what? The particularly sequence. So according to the sequence, if I consider here, what I am going to find out? The prothallus stage, first what they have mentioned, then meiosis in spore mother cells, fertilization, formation of archegonia and antheridium in gametophyte, then transfer of what? Anthrozoids to the archegonia in presence of what? Water. So this we have to arrange it by sequence wise. If I consider first what can occur here with respect to this option only, what we have to imagine, right. So meiosis 1 in the what? Spore mother cells. Meiosis in spore mother cells, that will be the what? First one. Then next what I am going to see here, that is what? Prothallus stage, right. That is what? Second one is a prothallus stage. Then the with respect to the thallus only, what I am going to see? The formation of archegonia and antheridium in a gametophyte, right. That is what actually the third one. And the fourth one if I consider, which is nothing but what? Transfer of anthrozoids to the what? Archegonia in presence of water, that is the fourth one, right. Then the particularly here if I consider fertilization, later lastly what I am going to see? The what? Fertilization, right. So that is what? The fifth one. So according to this, if I arrange, what I am going to get here? The B, second one is A, third one is here the particularly D, and the fourth one is the particularly E, and the fifth one is nothing but the C, right. So B, A, D, E, C. So that is, if I see this, the option 1 is what? Correct, right. That is what? B, A, D, E, C, right. Then Moving on to the what next here. Cardiac activity of the what heart are regulated by. Cardiac activity of the heart is actually regulated by. Here majorly it is regulated by means of what? The nodal tissue, a special neural central in the particularly medulla oblongata, adrenal medullary hormones and adrenal cortical hormones. So according to that, if I consider here the nodal tissue is required, then the something called as what? A specialized neural central in the what? Medulla oblongata, then the adrenal medullary hormones. So here the particularly option 1 is what? Correct. Here option 1 is correct, right? Then moving on to the next, right? Which of the following organisms cannot fix what? Nitrogen. So they have given what? Different options. Who is not going to carry out what? Fixation of what? Nitrogen. Azotobacter, yes, it carries something called as what? Nitrogen fixation, as it is being present with the rhizosphere. Oscillatoria you can find out with respect to the paddy field, lodging. So there also it carries what? The nitrogen fixation. Anaben and nostoc, these are the what? Majorly cyanobacteria. So even oscillatoria if you consider, right? So known to be the what? Cyanobacteria, which are meant for what? Carrying out specializedly nitrogen fixation, and they have a specialized component, that is what? Heterocyst, what we call it as. So here the one option left is, that is what? Volvox. Volvox cannot carry out what? Nitrogen fixation, that is what? Option D, and it is related with respect to the what? Algae, right? Then transfer, next is what? The statement based. Transfer of RNAs and ribosomal RNA do not interact with what? mRNA, right? Transfer of RNAs, that is transfer RNAs and ribosomal RNA do not interact with mRNA, they are saying. Statement 2 is saying RNA interference takes place in all eukaryotic organism as a method of what? Cellular defense, they are saying, as a method of what? Cellular defense. Now when there is a protein formation, right? Protein formation, what we know is DNA will pass on its message to the what? mRNA, right? mRNA will definitely come in contact with what? Both the particularly tRNA as well as what? The rRNA, right? But here they are saying so that it can produce what? The protein, so that it can produce what here? The protein. But they are saying here transfer of RNAs and ribosomal RNA do not interact with the mRNA. They do interact with what? mRNA to carry out what? Protein synthesis. So this statement is wrong. And RNAi interference, or we call it as a gene silencing method, adopted by means of what? Specially eukaryotic organism as a cellular defense mechanism, right? So there if I consider this option is what? Correct. So here according to this, statement 1 is incorrect and the statement 2 is correct, that is option 4 here, right? So option 4. Okay, we were with the question, that is 115th question. Here they have given one type of a diagram, and that is related with respect to the what? Plasmid, and we already know the plasmid can act as a what? A vector, so that which carries the gene of what? Interest. So here they have mentioned something called as what? Beta galactosidase event diagram was, if you observe. Then they have mentioned tetracycline, ampicillin, right? Then with respect to that only I am going to see here what question says. In the above represented plasmid, an alien piece of what? DNA is inserted at what? EcoR site, right? That is EcoR1 site. Which of the following strategies will be chosen to select the what? Recombinant colonies? Will be chosen to the what? Select what? Recombinant colonies. So using ampicillin and tetracycline containing medium plate, they are saying. Then the blue color colonies will be selected, white color colonies will be selected, blue color colonies grown on what? Ampicillin plate can be what? Selected. So overall with respect to this I have to find out what the correct answer, right? So what they are saying, at this particularly segment where which is actually already coding for what? Here beta galactosidase. Here it is coding for beta galactosidase, right? And that gene itself we are going to remove, and we are going to add the foreign or alien DNA into that particularly segment, right? So when I'll add, what will happen? This beta galactosidase activity will get what? Nullify, right? Which is not going to code for what? Beta galactosidase enzyme now, right? So there my gene of interest will go and get inset, right? Now this is purely an example. By seeing this, this is a purely an example of what? Insertional inactivation, right? Insertional inactivation. So what we know, the colonies, consider if with respect to the what? Petri plate, the colonies, and I have supplied what? Lactose into the particularly media. The recombinant colonies where we have added our alien DNA, that will not have the property of what? Beta galactosidase, right? So there itself, that is what we have inserted, and we have inactivated the beta galactosidase gene. Now when there is a lactose into the media, the colonies, if you consider, which appears what? Blue, right? They are what? Known to be the non-recombinant one. They are known to be the what? Non-recombinant one, right? Reason for it is, like, if consider it produces something called as what? The particularly beta galactosidase. That beta galactosidase will react with lactose, convert that lactose into what? Glucose plus galactose, and this galactose will react with one more chromogenic substrate what we have added, that is nothing but what? X-gal, right? That is nothing but what? X-gal. And when it react with the X-gal, the colonies becomes what? Blue in color, right? Where I have added the particularly gene of interest, there it will not have beta galactosidase activity, and it will not convert the lactose into what? Glucose and galactose. Now galactose will not react with X-gal, then there will not be any like what? Blue colored colonies. So my recombinant DNA or my recombinant plasmid is present inside the bacteria with what? White color colonies will be what? Selected, and that itself is what? The recombinant one. That itself is known to be the what? The recombinant one. So your option is what? 3, right? Then moving on to the what? Next question. Which of the following genetically engineered organism was used by Eli Lilly to prepare a human insulin, right? So they have mentioned what? Bacterium, viruses, yeast, mate fats, right? So here if you consider, they have chosen the bacterium, right? Genetically engineered bacterium. Majorly which one? The particularly E. coli. Why we are calling it as genetically engineered one? Because the mainly insulin forming gene, which is actually isolated from beta cells of pancreas, right? Beta cells of pancreas is inserted into the particularly plasmid of the particularly E. coli, right? So here Eli Lilly has chosen the bacterium, right? They have chosen what? The bacterium to carry out what? The production of what? This particularly recombinant insulin, right? So here option 1 is correct, right? Then moving on to the next, that is what? Name the class of an enzyme that usually catalyzes the following reaction. So reaction wise, if you consider what? Here SG is a combination and S hash is without a combination. Then what has happened? S became to what? Without combination with respect to the enzyme, and S hash and G got what? Combined here. So what it means? It means the particularly, and even what they are saying, G is what? A group of what? Other than hydrogen. S is a substrate and S hash is another substrate. So based on that only, what has happened here? The transfer of this particularly group took place, and that is G is nothing but what they have mentioned? A group other than the hydrogen they are mentioning. So other than the hydrogen means what? We are going to call it as transferase. So here the option 3 is correct, right? So which actually transfers something called as what? A group to the what? Next substrate, and that can be carried by means of an enzyme called as what? Transferase here, right? Then moving on to the next question, that is find the statement that is not correct with regarding to the what structure of what monocot stem, which is not correct with respect to the what structure of monocot stem. What they are mentioned here: hypodermis is parenchymatous, vascular bundles are scattered, vascular bundles are conjoint and closed, phloem parenchyma is absent. So according to that, if I consider here, vascular bundles are scattered in monocot, right? In then vascular bundles are conjoint and closed one, that is even with respect to the monocot. Hypodermis is a parenchymatous. This is not we are going to find out with respect to the what, the monocot stem. So here the option 1 is what correct. Then phloem parenchyma is absent. Yes, with respect to the monocot stem, phloem parenchyma is absent. Now which is not related with monocot is hypodermis is parenchymatous, right? Then moving on to the what next question here, that is the correct sequence of event in the life cycle of what bryophyte is, right? So current sequential event what we have to find out. So according to that, if you consider the mentioned first one as fusion of what anthrozoids with egg, attachment of what gametophyte to the substratum, then reduction division to produce what haploid spores, formation of sporophyte, then release of what anthrozoids into the water. So first what will happen, attachment of gametophyte to the what substratum, that is here first one, right? Then second what I am seeing here, the release of what anthrozoids into the what water, right? That is second one. Then the fusion of what anthrozoids with the egg, that is what the third one, right? Then the particularly fourth one is formation of sporophyte again, so that is what the particularly fourth one. And the particularly fifth one is nothing but what, reduction division to produce again haploid spores. So this is what actually the correct sequence. So sequence wise if I write an option, if you consider, that is first one is what B, right? Second one is what E, third one is that is nothing but what A, right? And fourth one is D, and the fifth one is what C. So here I have to find out the option, that is B, E, A, D, C, right? So here B, E, A, D, C, that is what option 3 is what correct, right? So option 3 is correct regarding this. Then which are the correct, right? Which are actually statement correct, right? That what we have to choose. Computed tomography and magnetic resonance imaging detect cancers of what internal organs. Then chemotherapeutics drugs are used to kill non-cancerous cells, right? Then alpha interferons activate the cancer patients, right? Then the immune system and help in what, destroying the tumor. Then chemotherapeutic drugs are biological response modifiers, right? Then in case of what leukemia, blood cell count are what decreased. So based on that, what we have to find out, we have to find out the correct one, that is nothing but what, the computed tomography and magnetic resonance imaging detect the cancers of what internal organs, that is what option A, right? And then one more is what, alpha interferons activate the cancer patient immune system and help in destroying the particularly tumor, that is what A and C is what the correct here according to this particularly what question, right? So A and C is an answer, right? Then moving on to the what next, match the column. They have mentioned what the particularly centromere, cilium, then the cristae and cell membrane. So we have to find the what here, the match the particularly column. Then centromere is related with what, related with the cell division, right? Then cilium is related with respect to the what, cell movement, right? Then cristae that can be found out with respect to the what, mitochondria. Then cell membrane is related with what, phospholipid bilayer, right? So according to that, if I consider, the option 4 is what correct here, right? Then moving on to the next, that is what, different pigments given and we have to compare, we have to see which color does it looks, right? So according to that, if I consider here majorly that chlorophyll A, if I consider, chlorophyll A is related with what, blue and green color. Chlorophyll B, if I consider, it is actually related with what? Yellow green color, right? Then the particularly chlorophyll, xanthophyll is what? Actually the yellow color, right? And the lastly the carotenoid is nothing but what? It is related with what? Yellow to yellow orange color, right? So here the option 2 is what? Correct, right? So match the column, right? Match the following, that what? So here option 2 is correct regarding this. Then find the correct statement, right, related with what? Pregnancy they have mentioned, and correct statement basis we have to actually find out, right? In human pregnancy, the major organ systems are formed at the end of what? 12 weeks. Then in human pregnancy, the major organ systems are formed at the what? End of 8th week, right? So according to this, if I consider these two statements, A is what? Correct, but B is what? Wrong, right? So in human pregnancy, the major organ systems are formed at the what? End of 8th week, they are saying. Actually the beginning of what? Heart beat, you will find out with respect to the what? The particularly 8-week period, right? So there majorly, so like that of a development, but there will not be any like what? Major organ systems are formed at the end of what? 8th week. So just fetus is developing, trying to get what? Cope up, right? Then in human pregnancy, the heart is formed after 1 month of what? Gestation. So after the 1 month of gestation only, we are going to get what? The formation of what? Heart. That what I have told you, 8 weeks, right, related to that itself. So here if you consider the option C is what? Correct. In human pregnancy, the limbs and digits develop by the what? End of the 2nd month. Yes, that is what? Even correct. Then in human pregnancy, the appearance of what? Hair is usually observed in 5th month. Yes, that even can be seen. So according to this, choose the what? Correct answer from the what? Option below. So correct one are what here? The A, then C, then D and E, right? So that is what? Option 4 is what? Correct here, right? Then moving on to the next, that is what? The seed of cereals, the outer covering of endosperm separates the what? Embryo by a protein rich layer called as, right? If I consider the monocot seed especially, the layer which is actually going to separate something called as what? The outermost we call it as seed coat, right? The now inside this I am going to find out what? Endosperm, and the embryo is going to be what? Separated. So that separation can be seen with what? This specific layer, and this specific layer we called it as alveolon layer, right? And this alveolon layer itself is proteinous layer, right? Or it is a protein rich layer, that what we are going to see, right? So such kind of questions we have already discussed with even what? Mock test, right? So that is what? The option 4 is what? Correct here. That is what? Alveolon layer, right? Then moving on to the next, that is which of the following diagram is correct with regard to the what? Proximal P and the distilled tubule of the what? Nephron. So proximal tubule, proximal cannulated tubule and distal cannulated tubule. So we have to find out what is the correct match, what exactly and how it can be matched with respect to this. So by seeing all the diagram, what I know is the option 2 is correct, diagrammatic basis, that what? The intake of ammonia, water and giving of what? Bicarbonate, NaCl, water. Then that all things what you are going to find out with respect to the what? This diagram, and that is proximal cannulated tubule and the distal cannulated tubule. So here option 2 is what? Correct. Then moving on to the next, that is identify the part of what? Bioreactor which is used as what? Foam breaker, right? From the what? Given figure, right? So we have to find out what? The correct foam breaker, right? So foam breaker is related with respect to the what here? The particularly C, which is known as what? The foam breaker, right? It is a foam breaker. But the B is nothing but what? It's actually motor, right? A is nothing but what here? It is a impellers, right? And here it is what? The outlet, right? Inoculum what we are going to collect it from. So here according to that, the foam breaker, that is what? Option C, that is what option 4, that is nothing but what C, which is known to be the what foam breaker here, right? Then moving on to the next, that is given below are two statements. One is labeled as assertion and one more is what reason. A typical unfertilized angiosperm embryo sac at maturity is 8 nucleate and 7 celled, right? Then the egg apparatus has 2 polar nuclei, right? Now typical typical embryo sac of a angiosperm, if you consider what it has the particularly embryo sac. At microfilar end we will find out what the particularly the egg cell or egg apparatus, right? Then at the center we will find out what the 2 polar nuclei, and at the chelazal end I am finding out the 3 antipodal cells. If I label it as like what here, 2 synergids, right, and with respect to that 1 egg cell, right. At the center I am finding what here, the 2 polar nuclei, right, and at the what 3 of them are what antipodal cells. So at maturity if I count it, it is nothing but what 8 nucleated and 7 celled, like that of what embryo structure what you are going to observe. So here they have asked related with respect to the something called as what the egg apparatus. These 2 of them, right, egg 2 synergids and egg apparatus, egg cell itself will make something called as what egg apparatus here, right. But according to the statement, if you see, the egg apparatus has 2 polar nuclei they are saying, right. So egg apparatus doesn't has what 2 polar nuclei, it has only like what 2 synergids and 1 egg cell. So according to this, if I consider here both the assertion and reasons are true, right, then reason is a what correct explanation regarding that. Then both the assertion reasons are true or reason is not the correct explanation. Here assertion is true but reason is what actually false, right. So reason is false, assertion is true. So here option 3 is what correct, right. Then moving on to the next, here a specialized membranous structure in the prokaryotic cell which help in the what cell wall formation, DNA replication and the respiration. If I consider the particularly a bacterial cell, consider it has a flagella, outermost layer we are going to call it as cell wall, right. Innermost layer we will find out some foldings over, right, and these foldings are majorly can be called as what the mesosomes, right. We call it as what mesosomes, and these mesosomes itself will help in what the particularly cell wall formation and the DNA replication and even respiration. So according to this particularly option, if I consider, the mesosome is the correct answer, right. Mesosome is a correct answer. They have mentioned endoplasmic reticulum, that is what actually absent over with respect to the word prokaryotic. Then cristae are part of what mitochondria, right. So that is even what absent. Then chromatophore, which is what the colored component what we are going to see. So according to that, if I consider the mesosome, that itself is a what membranous specialized structure which will form something, which will carry out what help in cell wall formation as well as the DNA replication and even respiration, right. Then moving on to the next, which of the following are the most post-transcriptional event in a eukaryotic cell, right. So following events, post-transcriptional event they are asking. So transport of pre-mRNA to the cytoplasm prior to the splicing, no, right. Prior to the splicing, if this particularly pre-formed RNA or majorly first formed RNA, if you consider, we call it as what hnRNA, if it moves out, then definitely what will happen, it is going to react with the other component. So priorly what splicing it won't go. Then removal of what introns and exons, introns and joining of what exons, yes, that can be seen. Addition of what methyl group at 5' end of the hnRNA, yes, that even can be seen. Additional of what adenine residues at what 3' end of the what hnRNA, yes, that can be seen. Then base pair of what? Two complementary RNAs, right? So according to that, if I consider here, that is B, C and D, right? Choose the what correct options with respect to that. B, C and D, if you consider B, C and D, that is what correct, but these all are what incorrect. Remaining what A and E are incorrect, related with respect to the what post transcriptional events, right? Then so here option 2 is what correct. Moving on to the next, what is the pattern of what inheritance for polygenic trait? For polygenic trait, what is the pattern of inheritance for polygenic trait? They are asking. Mechanism of inheritance pattern, non-Mendelian inheritance pattern, autosomal dominant pattern, and X-linked recessive inheritance pattern. So according to that, if I consider the pattern of inheritance for polygenic trait is known to be the what? Non-Mendelian inheritance pattern. That is known to be the non-Mendelian inheritance pattern, right? So here option 2 is what correct, right? Then moving on to the next, that is which of the following enzyme contains what? Heme as what prosthetic group? Heme as prosthetic group, where usually these kind of what enzymes carries what catalysis or catalase enzyme itself has something called as what heme as a prosthetic group. So that is what option 4 is what correct here, right? Option 4 is correct, right? So catalase has what heme as a prosthetic group with respect to it, right? Then they have mentioned something called as what each of the following characteristics represents a kingdom proposed by what Whittaker. Arrange the following increasing order of what complexity of what organism. So we have to carry out what arrangement of this particularly. So according to that, if I consider here multicellular heterotrophs with what cell wall made up of what chitin. Multicellular they are saying. Then heterotrophs with what tissues, organ or organ system level of body organization. Then they are saying heterotrophs and mainly organ system level organization they are saying. So correct order according to how we are going to arrange the Monera first, right? Following to that what actually Protista, right? And Protista to what fungi, and fungi to what plantae and animal, right? So like that of what the organization what we have to do one after the another, right? So like this of what arrangement, right? Then here what they have mentioned next is prokaryotic with cell wall made up of what polysaccharide, and amino acids. Eukaryotic autotrophs with what tissue or organ level body organization. Then eukaryotic with what cellular body organization, right? So here if I consider prokaryotic with a cell wall made up of what polysaccharide and amino acid, that I can find out with respect to the word Monera, right? Then eukaryotic with the cellular body organization, that I can find out with respect to the word Protista, right? Then fungi related, if I consider multicellular heterotrophs with a cell wall made up of, so that you can consider with respect to the word fungi. Then plantae what it left out, related with what eukaryotic autotrophs with a tissue or organ level body organization, so that is what here D, right? Then lastly what left is like here heterotroph with what tissue organ and organ system level of body organization, and that can be with what animal. So option wise is C, E, A, D and B. So that is nothing but what here C, E, A, D and B. So option 2 is what correct here. So option 2 is correct according to the pattern if I consider and compare the character, right? Then moving on to that, who is known as father of what ecology in India, right? So who is known as father of what ecology in India, that is Remedio Mishra, right? Remedio Mishra is known as what father of what ecology in India, right? Then here the like what a different scientist name and even eukaryotin, euchromatin and heterochromatin what they have mentioned, and with respect to that the characters given we have to what match according to. So Alfred Hersey and Martha Chase, what they have carried out? They have confirmed something called as what DNA as a genetic material by means of using what E. coli and bacteriophage, bacteria, right? Then euchromatin is majorly loosely packed and lightly stained, right? Because euchromatins are transcriptionally active, right? Because of that only they are loosely packed so that transcription process can be carried out. Frederick Griffith used what here? Streptococcus pneumoniae for his experiment, majorly Griffith transforming principle. Then heterochromatin, if I consider, which is nothing but what densely packed and darkly what stained. So according to that, if I consider here, the option 3 is correct, right? So here option 3 is correct, right? Then moving on to the next, that is neoplastic characteristics of cell refers to. Neoplastic characteristics of cell refers to a mass of what proliferating cell, rapid growth of cell, invasion and damage to the what surrounding tissue, and those confined to what origin location, right? So according to that, if I consider here, the A, B and C are the what the characteristics which can match up with respect to the what neoplastic, right? So that is a mass of what proliferating cell, then rapid growth of what cell we are going to find out, then invasion of what damage to the what surrounding tissue. That what I am going to find out with respect to the neoplastic as a characteristics of what cell. Alright, then given below what two statements here, they mentioned what the DNA fragments extracted from what gel electrophoresis can be used in construction of what recombinant DNA, right? Then statement 2 says smaller sized DNA fragment are observed near the anode while larger fragments are found near the what wells in a particularly agarose gel, right? So majorly it is related with what gel electrophoresis, right? So DNA fragments extracted from here, that here what I am going to find out, yes, with respect to the agarose gel electrophoresis is a separation technique where we are going to separate the fragment of what DNA. So that itself what correct here. Then small sized DNA, yes, smaller and low molecular weight molecules will move faster towards the anode. Why it is moving the anode towards the anode? Because anode is positively charged and DNA is what? The negatively charged, right? So definitely it will move towards positive charge, right? And whichever is the lighter, that will move faster, and whichever heavier, that will move what slower. Most of the time it remains nearer to the what well. So here the statement 2 is even correct. According to that, if I consider both statement 1 and statement 2 are correct, that is what option 1 is correct here, right? Then moving on to the next, that is what they have mentioned something called as what different components. With respect to that, we have to match particularly, right? So they mentioned here what adenosine if I consider, it is nothing but what nucleoside. Then adenylate acid, that is nothing but what nucleotide. Adenine is nothing but what the nitrogen base, and alanine is nothing but an amino acid, right? So according to that, if I consider here, option 3 is what correct, right? Option 3 is correct. Understanding? So like this of what component, right? So easiest one that you can answer it back, right? Then the consider the following here, the reductive division for the human female gametogenesis starts earlier that of the particularly male gametogenesis. The gap between the first meiotic division and the second meiotic division much shorter for the what male compared to the female. The first polar body is associated with respect to the formation of primary oocyte. Then luteinizing hormone surge leads to disintegration of the endometrium, onset and menstrual bleeding. So according to that, if I consider overallly, choose the what correct answer from the what options given below. So correct answer wise, if I consider here, that is what option 1, that is A and B are what true, right? But C and D is what actually false. Mainly it is related with respect to the gametogenesis, the formation of what male gamete with respect to the what male reproductive structure, and female gamete with respect to the female reproductive structure. So there majorly it is a question related with what human reproduction, right? That is option 1 is correct. Then all living members of the what class cyclostomata are, all living members of the cyclostomata are known to be the ectoparasite. They are known to be the what? Living member, they are saying. Living members of the class cyclostomata are what? Ectoparasites. They are what? The ectoparasites. Then given below are what two statements. One is labeled as assertion and the other is labeled as what? Reason. Right. Assertion says the primary functions of what Golgi apparatus is to package the material made by the what endoplasmic reticulum and deliver to the what intercellular target and outside. Right. Then vesicle containing material made by the what endoplasmic reticulum fuse with what cis phase of the Golgi apparatus and they are modified and released from the what trans phase of what Golgi apparatus. So we have to find out what the particularly assertion reason, how closely they are related and how that can be seen. So you consider here actually assertion and reasons are true and reason is correct explanation of what assertion, because after the formation of whatever the protein is, that is definitely going to move towards what Golgi apparatus and that towards is to the what trans way, right? So definitely that is what here both assertion and reasons are true and reason is the correct explanation of what assertion, right? Then moving on to the next, there here they have mentioned something sorry, moving on to the next, that is nothing but what they have mentioned again, like what column 1 and column 2 and we have to match according to that, right? So here if I consider majorly like that scutellum they have mentioned, scutellum you are going to majorly find out with respect to the here cotyledon of what the monocot, right? Scutellum can be called as the cotyledon of what monocot. The non-albuminous seeds if I consider rudimentary cotyledon, right? We are going to find out what here non-albuminous seeds if I consider that is nothing but what here the ground nut. Then epiblast is nothing but what rudimentary cotyledon. Perisperm is nothing but what persistent nucellus. Perisperm is a persistent nucellus. So according to that, option 1 is what correct. So option 1 is correct, right? Then moving on to the what next, given below are what two statements, that is what assertion and reason again. All vertebrates are vertebrates are chordate, but all chordates are not vertebrate. The members of what subphylum vertebra possesses notochord. During the what embryonic period, the notochord is replaced by a cartilaginous or bony vertebral column in adult. So according to that, if you consider here both assertion and reasons are true and reason is what the correct explanation of what assertion. So here reason is the correct explanation of what assertion, that is what option 1, right? Then identify the statement and that is not correct, right? Not correct we have to find out. Each antibody has what two light chain and two heavy chain. Yes, antibody is made up of what? One is what heavy chain and one more is what the particularly light chain, actually joined by means of what the disulfide bridges here, right? So next what they says, heavy and light chains are held together by disulfide bridge, right? It is held together by disulfide bridge, that is what correct. Antigen binding site is located at what C terminal of what antibody molecule, right? So they are saying what antigen binding site is located at C terminal regions of what antibody molecule, but it is present with respect to the what N terminal and that is what this option is incorrect, right? So here they asked something called as what the particularly which is not correct and that is what the option 3 is not correct, right? Option 3 is not correct, right? Then moving on to the next, splicing of a specific mRNA is possible via RNAi because complementary double stranded RNA, inhibitory ssRNA, complementary tRNA, non-complementary ssRNA. So according to that, if I consider here in a specific mRNA, the gene silencing can be carried out by means of what the complementary what double stranded RNA molecule, right? So that is what option 1 is correct, right? And this Right, then moving on to the next, in the R and Y, genes R and Y follows independent assortment. If capital R, capital R, capital Y produced round seeds, round yellow seeds, and small r produced wrinkled green seeds, what will be the particularly phenotypic ratio of F2 generation? Right, what they are saying is capital R, capital Y, it forming something called as what, round yellow seeds, right, round yellow seeds, right. Then the particularly small r, small y forming what, wrinkled green seed, right, wrinkled green seed. And what will be the what phenotype with respect to the what F2 generation they are asking. So phenotype with respect to the F2 generation, that is what direct question. Phenotypic ratio is 9:3:1, right. So here the answer is what, option 3, 9:3:1, right. Then moving on to the next, that is histones are enriched with. Histones are enriched with what these histones are? Histones are the specialized proteins which will be present with respect to the what, DNA, and together itself it will form what, the nucleosome, right. And this chain of what, nucleosome itself will form what, the chromatin network, right. So like this of what structure you are going to find out. Now they are asking what this histone is made up of, right, which type of what, the particularly amino acids. Here histone is actually made up of what, lysine and arginine. 60% of the histone molecule is consist of what, lysine and arginine. So that is what option 1 is what correct. Then moving on to the next, that is what the first menstrual cycle is called as. The first menstrual cycle is called as what, the menarche. At the beginning of the particularly menstrual cycle, at the age of what, 12 you consider, or during the what, puberty period or puberty period, we'll find out that is nothing but what, menarche, right. Then again match the column 1 with what, column 2. Here they have mentioned heart, kidney, gastrointestinal tract, then adrenal cortex, and they have mentioned something called as what, different hormones over there. So according to that, if I consider, right, according to that if I consider here, that is nothing but here heart, if I consider, heart is related with what, the particularly arterial natriuretic factor. Then kidney is related with respect to the what, erythropoietin. Gastrointestinal tract, if I consider, it is related with respect to the what, secretion, and adrenal cortex is related with respect to the what, aldosterone. So according to that if I consider here, that is what option 4 is correct, right. According to that if I consider here, the option 4 is correct. The match the following, right. Then the protein portion of an enzyme is called as, the protein portion of an enzyme is called as what, apoenzyme, right. That is nothing but called as what, apoenzyme, right. The direct form of what questions. Then which of the following is the unit of productivity of an ecosystem? The unit of productivity of an ecosystem. So rate of what, productivity can be measured in terms of what, kilocalories meter square per year, right, per year, that is what unit. Or else with respect to the gram basis if I measure, that is meters per gram meter per square per year, right. So that is what with respect to the what, gram basis. So here the option 4 is what correct, right. Kilocalories meter per square per year, that is what the option 4 is what correct, right. Okay, moving on to the next question, that is majorly here sweet potato and potato represents a certain type of what, evolution. Select the what correct combination of the what term to explain the evolution, right. So if you consider sweet potato and the particularly potato represents the particularly analogy, right, and they are known to be the what convergent type of what evolution, right. Homologous organs where the origination is same, but performs different function. Here origination is different and performs what same type of what functions. So here the according to that, if I consider the majorly like what option 1 is what correct, that is what analogy and known to be the what convergent type of what evolution, right? Then moving on to the particularly what with the help of what given pedigree, find out the possibility for the what birth of a child having no disease and being a particularly carrier, has the what a disease mutation in one allele of what particularly gene in the what F3 generation. So we have to calculate according to what mentioned x y x dot x, right? It is being what a diseased, and according to that if I consider overallly the answer here will be the particularly what 1/4, right? Answer will be here the 1/4, right? So based on that only like what pedigree chart we have to see how actually gene is passing, is it passing or is it skipping a generation, and then we have to what answer it back, that is what 1/4 of the particularly what child is going to suffer or have that particularly a disease, right? Or majorly like what possibility of a child having no disease and being a particularly what carrier. So here the option is 1/4, right? Then moving on to the what next question here related with what assertion and reason. Cells of the what tapetum possesses dense cytoplasm and generally have more than one nucleus, right? Then reason says the presence of more than one nucleus in the what tapetum increases the efficiency of what nourishing nourishing the particularly developing microspore mother cells, right? So according to that if I consider here assertion and reasons are true and reason is what actually the correct explanation, because the outermost layer of what this pollen sac which is known as what the tapetum, that provides nourishment to the what developing pollen mother cells, right? So there it can have what the dense cytoplasm and generally have more than one nucleus, so provides a proper nutrition to the what the pollen mother cells or microspore mother cells. So here option 1 is correct, right? Then how many meiotic and mitotic divisions are needed for the development of what mature female gametophyte from the megaspore mother cells in an angiosperm plant, right? So how many meiotic and mitotic divisions needed to occur for the development of mature female gametophyte from the particularly megaspore mother cells. So here mainly it should occur what meiosis 1 and mitosis should occur what the particularly here the 3 times, right? Mitosis should occur what 3 times, because with respect to the meiosis consider this is megaspore mother cell, it will undergo what meiosis overally and forms what the 4 cells. Inside that one cell will develop as what embryo sac. There it will have what the particularly one nucleus primarily. Then later it will undergo mitosis, right? Mitosis to form what 2 nucleus, one mitosis. Then later again mitosis 2, that is M2 I mentioned. Then there will be like what 4 nucleus, right? Then lastly what mitosis 3, right? Then it will have totally what 8 nucleuses, right? Because here if you consider overallly the matured female gametophyte or matured embryo sac will have 8 nucleus and 7 celled. Overallly here only the nuclear division is going to take place. So according to that meiosis 1 and mitosis 3 is a correct answer. Then which of the following is an example of what zygomorphic flower? So which of the following is an example of zygomorphic flower? That is nothing but what pea. Pea plant is known to be the what zygomorphic flower. Remaining of them are what the actinomorphic type of what flower. So here option 3 is what correct. Then moving on to the next, that is after maturation the primary lymphoid organs, the lymphocytes migrates for interaction with antigen to secondary lymphoid organs tissues like, right? So it will move towards what the secondary lymphoid organs right? So there if I consider overallly where it can move from what like what spleen, then you can find out with respect to the lymph node, and then you are going to find out what Pierce patches, because the primary lymphoid organs are what here thymus and bone marrow, right? And these of them are what secondary lymphoid organs. According to that, if I consider, the option 4 is what correct, right? Then moving to the what next question, that is what statement based. Fig fruit is a non-vegetarian fruit, as it has what enclosed fig wraps in it. Then fig wraps and fig tree exhibit the mutual relationship, as fig wraps completes its life cycle in fig fruit and fig fruits get pollinated by fig wraps, right? So here if I consider here statement 1 and statement 2 are correct. Again one more option can be seen here, that is statement 1 is incorrect and statement 2 is correct, right? Why it is being too complicated here? Because if you consider some of the species of fig are even what vegetarian one, right? But here they are mentioning what non-vegetarian. Some of them are what even non-vegetarian. So here the answer can be what 1 and 4, right? Then what is the main function of what spindle fiber during mitosis? So main function of spindle fiber during mitosis is to separate the what particularly chromosome. That is what the option 1. So spindle fiber, that which will going to separate the chromosome with respect to the what the division aspects, right? With the stages of even division. So to separate the chromosome is an correct answer. Then which of the following is the characteristic features of what gymnosperm? So characteristics features of gymnosperm we have to find out. So seeds are enclosed in fruit. No, it's a characteristics of angiosperm, right? Then seeds are naked. Yes, the seeds are naked because there is no any formation of fruit. That what we are going to find out with respect to the gymnosperm. Then seeds are absent. No, seeds are present inside the gymnosperm, right? Because of that only we call them as naked seeded plant. Then a gymnosperm have a flowers for the reproduction. No, they doesn't have what flower, right? So instead of that, majorly we will find out what a cone being present inside, male cone and female cone. So here the seeds are what naked is an a correct characteristic features of what the gymnosperm, right? Then moving on to the next, considering the following statement regarding the function of what adrenal medullary hormone, right? So functions we have to find out. Correct function. So here if I consider correct function regarding what, consider the following statement regarding the function of what adrenal medullary hormone, right? Here it causes what pupillary construction. No, it can't, right? And it is what hyperglycemic hormone. Yes, right? Then it causes something called as what the pillow erections, right? So that can be seen. Yes, it increases the strength of what heart contraction, right? That is what even I'm going to find out. So according to this, if I consider, the option 2 is what correct here, right? Then moving on to the next, why can't insulin be taken orally to diabetic patients? Because the insulin, if we take it orally, it will be what digested, right? And it will be digested in the gastrointestinal tract, right? It will be digested inside what gastrointestinal tract. Because of that only we cannot take it orally. So here option 2 is what correct, right? Then moving on to the next, that they have mentioned pteridophyte, bryophyte, gymnosperm and angiosperm and gymnosperm, and examples what they have given. According to that, I have to find out a particularly answer over here. That is, if I consider overallly here the pteridophyte, example is pteridophytes is nothing but what salvinia. Then bryophyte example, bryophyte example is what polytrichum. Then gymnosperm example is what here, the particularly salvia, right? Then the gymnosperm example is nothing but what, the particularly ginkgo, right? So according to this question, if I consider, the option 2 is what correct, right? So match the following again, right? Then moving on to the next, who proposed that the genetic code for amino acid should be made up of 3 nucleotide, and that is given by means of what, George Gamow, right? That is given by means of a George Gamow, who gave something called as what, the genetic code requires the 3 nucleotides, right? And then only it can code for an amino acid. So genetic codes are what? A triplet in a nature, right? So triplet in a nature, and that will code for an amino acid. So this is given by means of whom? The George Gamow, right? Then moving on to the next, that is they have mentioned something called as evil quartet, ex situ conservation, lantana camera, and dodo. So we have to find out actually the correct match of this is, if I consider here majorly evil cortex is related with respect to the what, the cause of what, biodiversity losses. Then ex situ conservation, if I found out, that is nothing but what, cryopreservation here. Then lantana camera is related with what, the alien species invasion. Then dodo is nothing but what, it's an extension. So according to that, option 2 is what, correct, right? Then moving on to the next, which of the following hormone released from the what, pituitary and is actually synthesized in the hypothalamus? So which is actually synthesized in hypothalamus and released from the what, pituitary? If I consider here majorly, that is nothing but what, anti-diuretic hormone, that is nothing but called as what, ADH, right? So ADH itself is released from the pituitary and is actually synthesized by means of whom? Hypothalamus, right? That is what, ADH, option 2, right? Then role of the water vascular system in echinoderms, right? So what is actually the role of what, the echinoderms here? That is what, mainly water vascular system. So respiration and the locomotion, yes, that can be seen. Then excretion and locomotion, no, right? Then capture and transfer of food, that even can be seen. Digestion of what, respiration, no. Digestion and respiration. Then digestion and excretion, no. Combination wise if I consider, so here the A and C are what, the correct one, related with what, water vascular system, and the mainly role of water vascular system in echinoderms. That is what, A and C is what, the correct, that is option 2 is what, correct here, right? Moving on to that, next is, which of the following type of what, immunity present at the time of what, birth and is a non-specific type of what, defense in human body? Direct question, that is nothing but what, innate immunity. Inborn immunity itself is called as what, innate immunity, right? So here option 1 is 2 is correct, right? Then moving on to the next, in bryophytes the gamma cup help in which one of the following? The gamma are known to be the what, which will lead into the formation of spores, and it is a structure formed to carry out what, asexual reproduction here, right? Asexual reproduction. So according to that, if I consider here, the option 2 is what, correct, right? So option 2 is correct, right? Then moving on to the next, in frog the renal portal system is special venous connection that act to link, right? And that is act to link with respect to the what here, kidney and the lower part of the body, right? So in frog the renal portal system is a special venous connection that acts to link with respect to the what, kidney and lower part of the particularly what, body, right? That is what, option 4. Then here they have mentioned what, statement based questions. In the ecosystem there is a unidirectional flow of what, energy of a sun from the what, producer to the consumer, right? Then ecosystems are exempted from the what, second law of what, thermodynamics. According to this, if I consider here, the statement 1 is correct. The flow of energy is a unidirectional, which will move towards move from sun to the what, producer to the consumer. So that is what actually the correct. Ecosystems are accepted from the what, second thermodynamics law? No, it is related with what, the second thermodynamics law. So here the particularly option 3 is correct, that is statement 1 is correct but the statement 2 is incorrect, right? So option 3. Then moving on to the next, which of the following statement is about, right? Which of the following statement about the ribisco is true? Ribisco enzyme they are asking, right? So it is active only in the dark. It has a highly affinity for the what, oxygen than the carbon dioxide, and it is an enzyme involved in the what, photolysis of water. it catalyzes the carboxylation of what? RUBP. So here, according to that, if I consider the option 4 is what correct, right? Because the RUBP mainly like what? Kelvin cycle. Inside that we know that ribulose 1,5 bisphosphate, 1,5 bisphosphate, right, in presence of ribisco enzyme, right, in presence of the majorly ribisco enzyme, it carries what? Carboxylation, that is what? Carbon fixation, to form two molecules of what? PGA, that is nothing but the phosphoglyceric acid, right? So there it plays very important role, that it catalyzes the carboxylation of what? RUBP, right? So here answer is what? The particularly fourth one. Then which of the following enzyme are not essential for the what? Gene cloning. They are not essential for the what? Gene cloning. If I consider here, that is what? Restriction enzyme is required for the gene cloning, yes, right? Then if you overallly consider, right, what is not essential they are asking. So for the gene cloning, restriction enzyme is required, right? Then with respect to that, if I consider DNA ligases is required, DNA mutase is not required, right? Then DNA recombinase, even that is not required. DNA polymerase, yes, it is required. So according to that, if I consider which is not essential they are asking, so here option C and D only, that is what option 1 is correct, right? Then moving on to the next, that read the following statement on what plant growth and development they are asking, right, and choose the word correct with respect to the word options, right, which one is actually the correct, right? So correct form 1 we have to choose, right? So parthenocarpic can be induced by auxin, yes, right? Then a plant growth regulator can be what? Involved in promotion as well as inhibition of what? Growth, yes, that can be seen. De-differentiation is a prerequisite of what? Redifferentiation, yes, right? Then abscisic acid is a plant growth promoter, no, right? Then apical dominance promotes the growth of what? Lateral bud, no, right? So here if you consider overallly, that is nothing but what? Here A, B and C are the what? Only correct related with respect to this, right? So that what we can find out, really choose the word correct option with all correct statements, right? So that can be what? Seen, and that is what option 1 is what? Correct here, right? Then moving on to the what? Next here, that is which factor is important for the particularly termination of what? Transcription. So majorly rho factors are known to be the what? The termination factor. Rho is known to be the termination factor, right? So here the rho factor itself is a what? Termination factor, and which it terminates the transcription as well as even what? Translation. That is what the option 3 is what? Correct here, right? Then moving on to the next, frog respires in water by skin and buccal cavity, and on land by the particularly skin, buccal cavity and lungs, right? So choose the what? Correct answers from the what? Following. So according to that, if you consider here, the statement is what? False for what? Water, but the true for what? Land, right? So statement with respect to the what? Skin and buccal cavity with respect to the what? Water, and land by what? Skin and buccal cavity and lungs, that you can find out. So the first statement is what? False for the what? Water, but true for the what? Particularly what? Land, that is option 3 is correct. Then moving on to the next here, twins are born to a family that lives next door to you. The twins are the boy and a girl. Which of the following must be what? True for them? They are monozoic twins, they are fraternal twins, then they were conceived through what? In vitro fertilization, and have what? 75% identical genetic content. So according to this, if I consider, they are known to be the fraternal twins, that is what option 2 is what? Correct here, right? Next here, which of the following microbes is not involved in the preparation of what? Household products? Which is not involved in the preparation of what? Household products? That is, they are what? Aspergillus niger, no, it is not, right? The lactobacillus, yes, it is used in household product. Then you consider Trichoderma polysporum, this is not used. Saccharomyces cerevisiae used, Propionibacterium ceremisiae is used, because lactobacillus is used to prepare what, curd, right? Saccharomyces cerevisiae is used with respect to the what, bread here, right? Then the particularly Propionibacterium ceremisiae is used in the preparation with respect to the what, cheese, right? So here the option 2 is what, correct, right? So that what we are going to find out. Aspergillus niger is used for the preparation of what, acids. Trichoderma polysporum is used for the what, the treatment of what, like what, tablets formation. Then they mentioned what here, match the least one with what, least two. So mentioned what, progesterone, relaxin, melanocytes, stimulating hormones, and the catecholamines, right? So that what they have mentioned. So according to that we have to what, match the column 1 here, right, with what, column 2. So if I consider overallly, progesterone is related with respect to the what, corpus luteum, right? Then relaxin is related with respect to the what, ovary. Then melanocyte stimulating hormones is are actually related with respect to the what, pars intermedia, right? Then catecholamine is related with respect to the what, adrenal medulla. So according to this, if I consider, the option 1 is what, correct here, right? Then moving on to the next, that is nothing but what, the blue and white selectable markers have been developed, and which differentiate the recombinant colonies from the non-recombinant colonies on the basis of their ability to produce color in the presence of what, chromogenic substrate, right? So what it says statement wise, the blue colored colonies have DNA insert in the plasmid, and they are identified as the recombinant one. Statement 2 says the colonies without blue color have the DNA insert in the particularly plasmid and are identified as what, recombinant colonies. So according to that, if I consider here, statement 1 is incorrect but statement 2 is correct, right? So majorly this is related with, as they have mentioned, chromogenic substrate. Here which chromogenic substrate is used? X-gal. And where this particularly technique can be seen, it is related with what, insertional inactivation. Already we have discussed in a previous question, insertional inactivation, right? So according to that here, the option 4 is what, correct, right? Now this X-gal react with respect to the what, lactose, and lactose which get divided into what, glucose and galactose, or digested by this beta galactosidase enzyme. Galactose will react with X-gal and forms what, blue colored colony. But non-recombinant will not have that particularly beta galactosidase production, and definitely because there itself we have added our gene of interest, then definitely it will be white in color. So here option 4 is correct, right? Then moving on to the next, that is the which of the following, right? So which of the following majorly equation represents the Verhulst-Pearl logistic growth population. So Verhulst-Pearl logistic growth population, how it is going to be represented graphically, if you consider, which has what, limited resource available, so only we mentioned it as the geographically it has what, carrying capacity. So limited resources available. So population, firstly it will go on increasing, but it get what, stabilizes over there, because it has what, carrying capacity that much, and that can be represented in the form of what, dn by dt, right, is equal to Rn, then k minus n divided by what, k, right? So this is what the Verhulst-Pearl logistic growth curve, right? So that is what option 2 is what, correct, right? So this is all about what the particularly NEET 25 question. We had a what, simple discussion on it. So please do watch it, subscribe our channel, like it, and try to what, improvise your knowledge too even. So thank you so much.